metabolism. What we're doing here is we're transitioning from all of the symbioses and the positive and negative interactions we've been talking about for the last couple of weeks. Now we're going to be talking about our module on sort of biogeochemistry, where we look at how microbes shape the planet in cycling different elements like carbon and nitrogen and sulfur. But we can't really get there unless we have a solid foundation of prokaryotic metabolism. And especially for the non-micro majors in the class, I like to do this lecture so that you get kind of a grounding. But I feel like for the micro majors, it's never a bad thing to have a nice review of some of the metabolic things. And I know some of you, most of you have learned some of this in general bio, et cetera, but we'll go over it. And of course, if you're taking bacterial physiology this semester, you're getting a lot more of this and in more detail with Dr. Amat than I give in this class. What I like to do is give this overview so that you understand kind of the diversity of the options that are out there. Because really that's the focus of this class, is sort of like microbial diversity and how those transformations are going on out in the environment. So the first thing, if you take nothing else from my class, we've talked about genetic diversity and species diversity, right? But just like with that, metabolic diversity is really the name of the game with microbes. If you think about what we do as mammals, right? From a metabolic standpoint, we're pretty limited, right? We eat carbon in some form, we break it down heterotrophically to get our food. But we're not photosynthetic, we're not chemoautotrophs, we're not able to be flexible, right? If you starve us for carbon, we're dead, right? Microbes, on the other hand, display a huge array of different metabolic capabilities, and in many cases, they can switch. So they have evolved over these billions of years they've been on the planet, the ability to be really flexible. And so, for example, you have some microbes that can be both anaerobic and aerobic, right? We can't do that, at least for extended periods of time. And so the other thing is there's lots of really cool stuff out there, like weird electron donors and acceptors, right, that we don't have access to. And so I like to use this lecture as a way to kind of give an overview. I'm not covering even a tenth of total metabolic diversity here. I just like to give you a sense of some of these things that you should think about. The other thing to think about is that these things, metabolic genes often move around via horizontal gene transfer. So in many cases, they are not what we call phylogenetically coherent, in the sense of this metabolism evolved once and it diversified within a clade or a group of microbes and just stayed there. So many cases, there have been movements across species, even across entire domains, of metabolic capabilities, but not always, right? So a classic example of something that evolved once and doesn't seem to have moved around much is methanogenesis, right? So this single group within the Euryarchaeota, they're the methanogens, they're archaeal, they do that, and they're the only ones that do it as far as we know. In contrast to that are the sulfate reducers. So sulfate reducing bacteria are commonly, that's a term that's commonly used, but we really need to say sulfate reducing prokaryotes, because it's likely that this evolved in the bacteria, but we know of archaeal lineages that have obtained the ability to reduce sulfate and use that for anaerobic metabolism. Okay, so those are two different contrasting examples of how overlaying these on the tree of life gives you a sense of how metabolism has evolved and how it gets around or doesn't. And I mentioned already the flexibility and the crossover, right? And so if you're a micro major, you've learned this, that something like E. coli, if you're in the presence of oxygen and you feed it glucose, what is it going to do with that glucose in the presence of oxygen? Metabolically, what would E. coli do with glucose in oxygen? This is not a trick question.
Will it break it down?
Yeah, how? What would it do? What process that you have studied extensively?
Aerobic respiration.
Exactly. Yes, that was not a trick question, but I think maybe, okay. But if we took that same E. coli and put it under anaerobic conditions, do they die? They don't, right? How would they break down carbon under anaerobic conditions? What processes occur anaerobically? And this is actually, Ashlyn, this was the correct answer that you put in the chat for this. They can ferment, right? So they can switch to a fermentative metabolism. Or if you've ever done a nitrate test, they can also respire NO3 and convert it to NO2, essentially breathing nitrate instead of oxygen. So E. coli is a classic example, but it is by far not the only one. There are many groups of microbes that can switch from aerobic to anaerobic metabolism, but this is particularly common among the facultative anaerobes. They can live in oxygen, they can live without it. Even among strict anaerobes, like sulfate reducers, though, some of them will switch from sulfate respiration to iron respiration. So there's a lot of flexibility that has evolved. So I'm going to walk us through different pathways that are important. And you don't have to memorize the formulas. I'll let Dr. Ramat test you on those things. What I want you to understand, though, is the diversity of these different processes and kind of how they're categorized. So carbon catabolism, right, which is the breakdown of carbon, right? Catabolic genes are breakdown genes. So the classic is aerobic respiration. And that E. coli that I mentioned, or any other facultative anaerobe, will want to be in the presence of oxygen to break down glucose, because look how much delta G you get out of it. And for those of you that aren't familiar with the delta G nomenclature, what word, you don't have to know it for this class, but what you need to know is it's good to be negative with delta G. That means you get energy out of stuff, okay? So negative almost 3,000 kilojoules per mole is a huge amount of energy, and that's why glucose respiration is so efficient and so favorable. By contrast, you get about 1/10 of that if you ferment the same glucose molecule. So you might ask, well, why do we want to do this if we could just do this? But remember, oxygen isn't always around. And in fact, as you, I hope, remember, on the early Earth, there was no free oxygen. So these kinds of anaerobic processes likely preceded aerobic respiration by billions of years or hundreds of millions of years, certainly. Okay. And so what you can see is that because the delta G is so much less, you get way less ATP out of it. And we're not going to go into the details. I'll let that be in physiology, right? But you have to put in ATP to get out. You put in 2 and get 4 out. And so it's a net of 2 ATP for fermentations, right? You probably remember, all of you have had glycolysis, which is the first step leading into either aerobic respiration or fermentation. So you probably remember that a little bit. For my class, what I want you to understand is there's an energetic difference here, but just because this yields less ATP and less energy doesn't mean it's not a viable lifestyle. And then the third option in terms of carbon catabolism are those anaerobic respirations. And this can be found in facultative anaerobes, like I mentioned, the E. coli, right? They can respire nitrate. But some of them, like sulfate reducers, are typically strict anaerobes, right? And they don't like to live in the presence of oxygen. They will be dead really quickly if you do expose them to that in many cases. For the respirers, many of them can use hydrogen as an electron source, right? So when you see an H, you should always be remembering, oh, if there's a proton, there's going to be an electron to go with it if I don't see a plus sign here, right? So hydrogen is actually a really good electron source, but you get way more electrons out of organic carbon where you have complex molecules like the glucose up here, right? Lots of electrons because there's lots of H's. But even simpler compounds like 2 and 3 carbon fermentation products like lactate can also be a good source of food. And so since they're not breathing oxygen, they have to use another oxidized substrate. So the key to this is to remember that you're going to look for oxygens in the molecule, whether that's sulfate or nitrate. They have those oxygen components, and so they are oxidized substrates. But the point is that even though they can do this anaerobically, just like with fermentation, the energetics are not as favorable. That said, you can still make a living being a sulfate reducer, okay? So this is a diagram. You do not have to memorize all of this, but I like to refer to this as the electron tower of power, right? And so I find that it's very confusing to understand how redox works, right? In general chemistry, you have to memorize Leo goes Gur or whatever the mnemonic device you did, right? But I know when I took general chemistry, I struggled with this concept, and metabolism relies so much on these oxidative and reductive reactions, but it gets confusing. So I like to just kind of have us take a step back and think about this. Up here, things are electronegative, and we know that because there's lots of electrons here, right? So you saw that with glucose, right? Tons of hydrogens, so there's tons of electrons that go with that, right? Obviously, oxygen, right, is the ultimate oxidizer, right? So it doesn't have electrons. It wants to steal electrons, right? Water does have electrons, of course. If you see the H, you know there's electrons there. But oxygen itself, right, is going to want to take and accept those electrons. And that's why oxygen evolved as this wonderful, really efficient electron acceptor. And so in general, if you donate electrons from things up here and you accept them with things down here, that's good. The difference or the spread or the farther they fall from where the electrons are coming from, strongly electronegative, and if they're being oxidized by these strongly electropositive elements, that's going to be good for energy. Okay, so you tend to want to pair things like glucose with oxygen or nitrate or iron. And that is how metabolism has evolved over these millions of years. Oxygen is great today, but it wasn't in abundance 3 billion years ago.
Dr. Dillon, you muted yourself.
You're still muted. I think the audio is just cut off somehow, Doctor.
Sorry, my headphones shut out on me. What I was saying is the things you'll notice here too are these are things like intermediaries for electron transport that you had to memorize when you were like Bio 212 or General Bio, right? So, cytochromes and those kinds of things, quinone pools and all that stuff you learned in other classes. Well, those are intermediates and they fall in between the end members. And so you can see how they can easily slot in as components as electrons are passed from one carrier to another carrier to another carrier to another carrier. And the reason that microbes do this is to get ATP, right? And who remembers if you have electron transport, like what you guys learned with like Krebs cycle, where does the ATP come from? How is that tied together with the electron transport? Who remembers? Why does moving electrons through a series of carriers end up generating ATP for, let's say, us? Or
Electron pumps from the mitochondria.
okay, and so in us, we do this in the mitochondria, but a bacterial cell can just do this at their membrane, right?
Yes.
And there's pumps, and what are they pumping again?
Electrons.
Well, the electrons flow, but what do they pump out? Protons. Yes. Thank you, Janice. Thank you, Angel. Thank you, Rika. Because remember, the ATPase, remember all that good stuff, right? ATPase, the protons get pumped out, and then they flow through the ATPase back into the cell or back into the organelle, which you learned in terms of mitochondria. But remember, a mitochondrion is just a symbiotic bacterium, right? So bacteria are going to be essentially doing the same kinds of things. Does that make sense? All right. So I'm going to give you a very quick overview of some of these different flavors of metabolic activities. So we've talked about aerobic respiration, and you've studied that before. So I'm not going to dwell on that. What you probably haven't studied before, unless you've covered this bacterial physiology yet, is anaerobic respirations, right? So that nitrate test that you guys learned if you were a micro major, right? The reagent turned red if the E. coli or whatever bug had done this. That's testing for this. Feed carbon, oxidize it with nitrate instead of oxygen, and convert it to nitrite. And this is catalyzed by the enzyme aptly named nitrate reductase. You are reducing the nitrate by one oxygen. We get rid of an oxygen. That means we put on electrons, right? So that's one way of doing this, and that's the simplest way, and that's what E. coli does. But there are other microbes that actually take it much further. So they go from nitrate to nitrite to nitrogen gas, okay? Or they go from nitrate to nitrite to other intermediates to ammonia. And all of these yield very good negative delta Gs. And if you have a strong negative delta G, that means you get lots of what out of it? What did I say a couple of minutes ago?
Energy.
You get lots of energy, and of course our energy currency is ATP. Right? Okay. Sulfate. So you'll notice that the delta Gs are negative, but they're much smaller. So if you're a sulfate reducer and you're oxidizing organic carbon, okay, this is just a couple of different ways of doing this. You can use sulfate or you can use elemental sulfur. Sulfate is more oxidized than sulfur, and so you get a little bit more negative here. The key is, are you negative? If you're negative, you can make a living as a heterotrophic breakdown catabolic organism, okay? Does that mean that nitrate utilizers are way better than sulfate reducers? No. It just means that these guys have to work harder to get the same amount of ATP. They're going to need to do a lot more respiration. But you can certainly make a living even with this fairly slightly negative. And then in between is another one. Here's iron. And I should point, I didn't point out. So here's oxygen. We know that's great. Here's nitrate, right? The spread is bigger between glucose and nitrate than it is between glucose and elemental sulfur or glucose and sulfate. So that's what I was referring to earlier. You want the difference in electronegativity to be bigger, and that tends to give you higher amounts, better delta G spreads, more electronegative, negative delta Gs. But it's not quite as simple as all of that. So for example, the delta G of ferric iron, which you might look at and say, oh, that should be even more than nitrate, is actually less. This is better than sulfate, but it's not quite as good. And that has to do with some of the complexities of the kinds of carbon that they're using. And you don't need to worry about that for this class, but still a good way to make a living. And if you think about it, this is totally weird. They're breathing iron, right? They're not breathing oxygen. They're not even breathing something like nitrate or sulfate. They're breathing iron. But yeah. So these are all catabolic reactions, okay? Where, and this is actually the opposite of rust, because this is taking oxidized iron and turning it into reduced iron, okay? These catabolic reactions are different, but you can turn those reactions the other way around as well. So instead of reducing things like sulfate and nitrate, you can oxidize them. And these are very commonly used by chemolithoautotrophs. So a chemolithoautotroph, when I say chemolithoautotroph, what's the thing that should pop into your head? Where do they get their carbon from?
Rocks, I guess.
So chemicals and rocks, right? So they get their energy from the rocks, but where do they get their carbon if they're chemolithoautotrophs? What's their source of carbon?
Carbon dioxide?
Exactly. So just like plants, right, or phototrophs, these guys are able to fix CO2. And that's what I mean up here at the top. The energy to do that doesn't come from the sun, though, right? If you're a plant or you're a cyanobacterium, you get your energy and your ATP essentially from the sun, right? We'll talk about those in a few minutes. These guys have to use rocks or chemicals as their source. They're essentially their source of electrons, okay, which can be used to reduce CO2, right? Which is a stepwise process where you have to be, reducing and adding electrons and hydrogens onto your carbons, right? So let's look at this. There's lots of different bacteria and archaea, for that matter, that do this, and they have evolved very complicated pathways, in some cases multi-enzyme pathways. But what you can see is that even though they're fixing carbon, the reason they can fix carbon is that these delta Gs are negative. And if you remember back to your photosynthesis, right, you need to get energy and then you spend that energy to do the hard work of carbon fixation. And that's essentially what's happening here. So if you oxidize something like hydrogen, which we know has electrons there, or sulfide, which has electrons, or iron. So this is the opposite of the previous slide. Before, I said that we were taking Fe3+ and turning it into Fe2+. That was iron reduction. This is iron oxidation or rust. So it's interesting because you have to think about these reactions will occur naturally in the presence of oxygen. But microbes have evolved enzymes, often multiple enzymes, often oxidase enzyme or monooxygenase enzymes. And these enzymes work faster than air, faster than the chemistry. The biochemistry has to be faster than the chemistry would be in terms of oxidizing these pathways so that the energy can be delivered to the microbe. Okay? And so you can see the broad array of different substrates that can be used, okay? And some of them are the exact opposite of what we said before. We talked about sulfate reduction. These guys are oxidizing sulfide back to sulfate. We talked about nitrate, right? In this case, you can take nitrite and go back to nitrate, okay? So these guys are doing the opposite reactions of what we saw before. Both sets of organisms make a living doing this, Okay. Often it is different organisms doing one versus the other. Usually it is. Okay. These reactions require oxygen. The other reactions, except for aerobic respiration, don't. Is everybody clear on that? Okay. Now, to make things even more interesting, many of these chemolithoautotrophs are facultative. So if you feed them some organic carbon, guess what they're going to do? They're going to oxidize their substrate, but then instead of using the ATP and reducing equivalents they get from that to fix carbon, they're just going to store it up. And they're going to get their carbon by breaking down organic carbon heterotrophically as well. So they can be both autotrophs and heterotrophs depending on the conditions. And so technically speaking, that would be referred to as mixotrophy. Evolution is wonderful. So these are just some pictures. So sulfur oxidizers have these cool granules. These are elemental sulfur granules that form. So this is a genus called Beggiatoa. They're long filaments and they store the elemental sulfur inside their cells. So they take sulfide and turn it to sulfur, and then they crawl around with the sulfur, and then later on they'll convert the sulfur to sulfate and they can continue to metabolize. And when you put a bunch of these together, they often form these really cool white streamers like this. Now this you might think looks an awful lot like rust. And again, this would happen aerobically in the air, but these are iron oxidizing bacteria that cause this to happen much faster. Okay. All right, so I know I've thrown a lot at you already. So let's have another poll question to make sure we're kind of all on the same page here. So which of the following would be associated with chemolithoautotrophy, what we just talked about? Would it be iron reduction? Would it be sulfur oxidation? Would it be denitrification, or D, all of the above? So I don't want to share my results. I want to restart, relaunch. All right, go ahead and answer it. Which of these would be chemolithoautotrophy? Think back to the previous slide and which of those would be examples from that? Right, is that everybody? Go ahead and log your answer. Okay. All right, so most everybody got it. So why is iron reduction not a correct answer? Does anybody want to take a stab at that? And one way to take a stab at it is, what would be the correct answer if iron was involved? Yeah, thanks, Jennifer. She put iron oxidation, and that's what Alessandra put as well. Right. So if we look back at the previous page, right? Get back to that, right? Right, iron oxidizers. But we have to go back to this page for it to be iron reducers. So these guys are not chemolithoautotrophs. They have to have organic carbon for their metabolism that somebody else gave them, okay? These guys, on the other hand, many of them can use organic carbon, but the reason they're called a chemolithoautotroph is they can all fix CO2, and they get their energy to do that from these oxidative reactions. So iron oxidation would be a correct answer, but that wasn't what was there. So the best answer was B, sulfur oxidation. I should point out the denitrification is an extension of that's what this is called, because you're removing nitrogen from the system. So nitrate goes to nitrite, goes to NO, goes to N2 gas. And because this is a gas that goes up into the atmosphere, the nitrogen is lost from the system. Okay. Farmers hate this, right? Because you convert their fertilizer into air. Nobody wants that. All right, is everybody on board with the poll question now? Okay. So now let's talk about photoautotrophy, or phototrophy more generally, because sometimes there's photoheterotrophy too. So thus far, we've been talking about breakdown reactions with like the reductive ones, but we did talk about chemolithoautotrophy, which can be anabolic reactions where you're taking CO2 and turning it into organic carbon. And so phototrophy is much the same way, right? So you have to use that energy. Instead of coming from rocks, though, the energy comes from light and all these specialized pigments. So as we learned earlier today, the delta G for aerobic respiration was negative 2,881 kilojoules per mole. So we know that you get lots of energy out of that, and it's going to generate a lot of ATP. Well, if you see a delta G that has a big plus number like this one, then you know you're going to have to put a lot of ATP into it to make this work. Okay. So there's two ways of phototrophy, and we're going to talk about both of them. So there's oxygen producing, and that's what you've learned about in other classes before, right? But as we've talked about earlier this semester, that likely was preceded by non-oxygen producing photosynthesis. So there are groups like the green and purple sulfur bacteria, the Chromaceaceae, those are the purple sulfurs. The Chlorobiaceae, these are the green sulfurs. And instead of water as their source of electrons, they use hydrogen sulfide. Still has electrons. How do you know? Because it's got these H2s here, right? So they take the electrons from here and they reduce CO2 to sugars. Okay. There's also microbes or phototrophs that do this without sulfur. And they can do this autotrophically, or in some cases, they serve as photoheterotrophs. What does that mean? If you're a photoheterotroph, if you have organic carbon around, you're going to use that carbon and you're not going to fix carbon. You're not going to do that dark reactions of photosynthesis. But you can still use your photopigments to generate ATP and use that for other cellular purposes, right? So those are anoxygenic, non-oxygen producing reactions. And then of course, the cyanobacteria and their ancestors and their close relatives, the prochlorophytes, these are the organisms that evolved the ability to use water as an electron donor instead of things like sulfur. And what that means is that there's O2 that comes out the other end. So we are eternally grateful for this because, of course, we breathe this stuff, right? But for the microbes, it's just a byproduct. So phototrophs have evolved very complicated suites of pigments for their metabolism. And the way this works is that different pigments, of course, have different colors, but the reasons they have different colors is because they absorb and reflect different wavelengths. So the reason that chlorophyll A, which is found in plants, but is also found in cyanobacteria, right? The reason chlorophyll A appears green is that it absorbs strongly in the blue and strongly in the red, but it doesn't absorb green wavelengths in the middle. It reflects them. So those trees out there, or that cyanobacterium, looks green color, but really the colors that matter are blue and red. Okay, and so different pigments have evolved the ability to absorb different wavelengths. There's other chlorophyll molecules referred to as bacteria chlorophyll. And what do you notice about these peaks here versus these peaks up there? How do these compare? Are they absorbing the same wavelengths?
It looks like they have like a higher absorbance as you get higher wavelengths, so higher energy wavelengths. I think like purple is one of the strongest, or maybe I have it reversed, but
Yeah, so it is backwards. So actually the higher energy are the lower wavelength ones, but you're absolutely right that they're absorbing up here. This is actually out into the infrared. It's not even visible anymore. Remember, visible goes to 700. But if you look at chlorophyll, it's absorbing red. But if you look at these, it's absorbing infrared. So it's a different wavelength. And even though these are lower energy wavelengths than these, you can still make a living there. So that's one of the things I'm telling you guys about today is that, you know, just because something is different and may yield less energy doesn't mean it's not worth using. The reason they absorb these different wavelengths is it all comes down to the molecule. And essentially what's different here is this side chain, right? So as you remember from chemistry, all you have to do is start monkeying around with some different moieties on a complex chemical, and they can do different things. So you get some differences in these different side chains, but particularly it's this, right? It's this yellow one. You change that, and suddenly it changes the spectrum. And you guys learned this in chemistry, right? You took chemicals and ran them through like spectrometers and chromatography, and you saw those differences, right? Essentially, microbes have just evolved the ability to exploit those chemical differences and how they are reflected in absorbing photons of light. And so it gets really complicated and interesting with all of these different R side groups. But basically, what it comes down to is there are many different molecules that have evolved. The bacteria chlorophylls have primarily evolved in non-oxygen producers. So cyanobacteria use chlorophyll A. But these purples and greens that I mentioned earlier, those anoxygenic phototrophs are the ones that have evolved this greater differentiation here. And the differentiation all comes down to the fact that in vivo or in real life, they absorb these different wavelengths. The reason this shows methanol is that a good way to know what pigment you're dealing with is to extract the molecule in methanol. And that changes the absorbance a little bit, but basically these are diagnostic. It'll always shift from about 805 to 771, but you can account for that. So what this allows is coexistence. And I always like to show this slide for this reason. So these are actually eukaryotic algae over here. So they're chlorophyll A, chlorophyll B containing algae living side by side with purple sulfur bacteria. Now, for the most part, the purple ones are going to be found underneath the green ones in this setting. And the reason that works is that these guys absorb different wavelengths, than these guys. So a photon of light coming through here, the portion of that photon that is absorbed by chlorophyll A will be stopped. But as the photon penetrates through the biofilm, okay, that's essentially what we're seeing here. those wavelengths will not be blocked, and then they are free to be absorbed by these sulfur bacteria. So this enables coexistence. That's really important in biofilms. Okay. Any questions about that? Okay, so again, you don't have to memorize these, but I like you guys to see this so that you can kind of wrap your head around this. So you guys learned about green plant photosynthesis, right? Do you guys remember, did they talk about the Z scheme when you guys learned about photosynthesis? Anybody heard that term before? Maybe, maybe not. Maybe they're not using that terminology at wherever you guys did your lower division courses. But basically, the thing to know about green plant photosynthesis, which of course is cyanobacterial photosynthesis, is that they have two photosystems, okay? And we'll see those in a second. But before there were two photosystems, there was just one. And there are still plenty of these anoxygenic phototrophs that live on our planet today that have a single photosystem. And so they have different pigments. We already covered that. And the pigments are going to be absorbing different wavelengths. So that's where the numbers come from here. 870 refers to the wavelength. And so in this case, this organism is absorbing the infrared light, and the light changes it from electropositive to strongly electronegative. And then the electrons, so it needs light and it needs an electron donor. And remember, the anoxygenic phototrophs usually get their electrons from things like sulfur. Okay, so we've got our sulfide, it donates an electron. That electron gets energized and strongly reduced at the reaction center, which is where the light photon hits. And then that electron can be passed from carrier to carrier to carrier to carrier. And you don't have to memorize quinone A and cytochrome BC. What I want you to focus on, the fact is that we're going from the strongly electronegative back down to electropositive. But it's like a water wheel, right? So if you spend the energy to get a bucket of water up on top of a water wheel, right? Gravity is going to want to pull that back down and do work, right? And when that happens, then guess what? We can generate electricity or something from it as human beings. This is much the same. The power of the light is being used to energize these electrons. And then as they flow back down, not by gravity, but by simply being passaged from one carrier to another, becoming less electronegative, more electropositive. As that happens, they can do work. And if you remember from your photosynthesis lessons earlier, right, the light reactions of photosynthesis generate ATP, and that can be used for a lot of purposes. But the other key thing that happens during the light reactions is we are going to generate reducing equivalents. Things like NADPH, FADH, NADH, right? And it's those reducing equivalents that go into the so-called dark reactions, which is where the carbon fixation happens, okay? So if you remember nothing else from this, I want you to be thinking about the fact that we're going to energize our electron. The electron then comes back down the cycle, and we use that to generate these so-called reducing equivalents. But as you can see from this, it's hard. And so there's actually a need to use a little bit of your ATP to do reverse electron flow. If you want to take the electron and stick it onto NAD or NADP, you need to spend a little bit of ATP to do that in this kind of organism, okay? So these are the purple bacteria. They got to spend money to make money, so to speak. But guess what? If you're photosynthetic, you've got ATP, right? So that's not really a problem, okay? All right, does anybody have any questions about this? So the goal of photosynthesis is generate ATP, but also generate these reducing equivalents like the NADH. So it turns out that there's not just one way of doing that. This is the purple bacterium that I just showed you. But if we look at some of those other anoxygenic phototrophs, they're not using water, they're not producing oxygen. They have different pigments, we already covered that, but they're also using different intermediaries, but they all have a single photosystem. And the greens and helios don't have to do this reverse electron flow. And the reason is, if you notice, they start off a little less electropositive, and that single photon makes them a little more electronegative. So it's like that water wheel is reaching up a little bit higher to begin with, so it can do the work it needs to do. And so in this case, what happens is the electrons get dumped onto, like the NADH, higher up. And so they don't have to spend the ATP to get them back up to that level, okay? Incidentally, this heliobacterium is a very rare bacterium found in hot springs. This organism was discovered by my PhD mentor, Dick Kastenholz, in a hot spring in the southern part of Oregon. And these can be found in hot springs around the world, but it just shows that even over the last 30 or 40 years, we discovered these novel things in the microbial realm. This is like discovering an entire whole suite of new animals, right? I mean, this is a huge kind of thing, even though it seems very innocent and maybe it's found in this one location. Anyway, so those are the phototrophs that are using sulfur, okay? But there are some that are so-called non-sulfur bacteria, which is a little confusing because many of these guys can use sulfur as an electron donor. But as I mentioned earlier, they can also skip that entirely and not fix CO2 if they have organic carbon in their environments. Now, these green non-sulfurs, these are things like flexibacteria, chloroflexus. These are often found in close association with other phototrophs like cyanobacteria. And so if they're found with those other phototrophs, they might get the organic carbon by mooching it off of carbon leaking from some of the carbon fixers nearby. In that case, then they can use ATP and they can photosynthesize, get ATP, but they don't have to fix their own CO2. So again, flexibility and adaptability. All right, so that was the single photosystem anoxygenic phototrophs, okay? But then, you know, about 2.5, 3 billion years ago, oxygenic photosynthesis evolved. So what you notice is that there's 2 photosystems, and that requires 2 photons of light, okay? And this is what I was referring to earlier as the Z scheme. If you turn it sideways, it looks like a Z. You could also call it the N scheme, I suppose, but that's not what people did. So instead of sulfide as the source of electrons, we have water, okay? One of the problems with this is that water donates its electron at an exceedingly electropositive state. And so you really have to have 2 photons of light and 2 photosystems, because what you're doing is you excite it, but it doesn't go high enough to do what we need it to do. So it starts the job it passes through these electron carriers, but it takes a second photon of light for it to go from here, which is high enough then to do what we saw in the earlier slides, which is produce reducing equivalents. The other thing that you can do is if you don't need to fix carbon, but you just want to generate energy, you can do what's called cyclical electron transport. So you can energize your electron, bring it here, energize it again, and instead of dumping it onto NADPH or NAD or NADP, right, you can just cycle it back from this ferredoxin molecule. It can come back over here and get re-excited and go back here. And what that allows you to do is build up like a cyanobacterium can build up ATP stores. And that's important in certain contexts. Okay. All right. Now, you might also be confused. Why is this called photosystem 2 and this is photosystem 1 when this comes in this is the first one and this is the second one? That's because the biochemists named these things and characterized them before we knew how they went. So the first one described was this one, so it was called photosystem 1. Then we described this one, but in reality, this one comes first. So just be aware of that. Now, interestingly, some cyanobacteria can shut off this part and just run photosystem 1. And that may be a holdover from their early evolutionary times, back when they may have evolved from an ancestor that had a single photosystem. So if you take a cyanobacterium and you put it under anaerobic conditions with hydrogen sulfide, they can do this. All right, so I just told you that you have to have 2 photosystems to make the Z scheme work for cyanobacteria. So my question is, why do that? What would be the advantage of using water as an electron donor then? Is it because it releases oxygen? Is it because water is readily available? Is it because sulfide can be toxic, or that water has higher energy electrons? What do you think the best answer would be? Why did cyanobacteria evolve the use of water as an electron donor? All right, any last answers? Okay. All right, so we're kind of all over the map on this one. So it's good that I asked that. All right, so it releases oxygen, which is true, but from the cyanobacterium's perspective, do they care about that? I see you shaking your head no, Victor. In fact,
They just want the electrons, right?
Right, they just want the electrons. And in fact, oxygen is hazardous. So all those free radicals we talk about, and the reason you should drink your pomegranate juice or eat your blueberries, right? Because they're full of antioxidants. What are we talking about there? We're talking about oxygen, right? We're talking about things like superoxides, singlet oxygen. These are hazardous to DNA and cell components. And so if you're a phototroph producing oxygen, that actually makes things a little bit risky for you. And so they have evolved things to deal with that. So for example, the carotenoid pigments, which you may have noticed on some of my earlier slides, those carotenoids likely evolved as a way of dissipating the oxygen radicals. And that's why we eat blueberries full of carotenoid pigments as antioxidants. So A is actually not a correct answer. B is the best answer, and that's what not a majority, but the largest number of you put. Water is everywhere, right? Water is life, and so it's a really great source. Sulfide is not everywhere, although it was a lot more common on the early Earth before we oxidized the atmosphere because of these critters. So, but sulfide was not, it can be bound up in metals and things like that. So that's likely one of the motivations, right? Sulfide can be toxic, but the microbes have evolved the ability to deal with that. And water has lower, right, less electronegative. So that's actually bad. And that's why we need the Z scheme, to make them electronegative enough to be able to reduce those NADs and FADs and things like that. All right, let's see. Okay, let's have a quick breakout. Let's see if I can do that really quickly. We will do a quick breakout. What I want you to do is talk about, look at this diagram, and it's in your slides, and think about how we may have gone from the stuff on the right, single photosystems, to the stuff on the left. So let me make it quick. We'll just do 4 breakout rooms. And I will move to 2. All right. So just take a couple of minutes and talk to your neighbors about how we might have gone from the stuff on the right to the Z scheme that I just taught you about. Marwa, are you able to join your breakout room? All right, so who wants to volunteer to share maybe what your group talked about? What do you notice about these things on the right, and then how they might have evolved to what's on the left? Anybody wanna?
I think that photosystem having like the two component one has two functions. Like one of them is storage of ATP. I
Okay,
think that is being done in single photosystem. So
okay.
that might be a to evolve into.
Did anybody notice, like, if you look at the specific carriers and things on the right-hand side, does anybody notice how those might kind of connect? What I'm getting at is, you know, if we were going from the things on the right to the thing on the left, how might that jigsaw puzzle be put together?
Is that something to do with horizontal gene transfer?
Potentially, absolutely. Or potentially gene duplication events, right? But what do you notice? Like if you look here, right? Look at this iron sulfur, ferridoxin. Do you see that anywhere on the left?
I think I see it.
Right? Do you see it here?
Yeah.
So if I look at this, I see these as being very analogous to photosystem 1. And then if I look at photosystem 2, it's not quite the same, but you know, you've got some quinones here. We've got quinone, you've got cytochrome. So one of the thoughts that's out there is that if we categorize these guys, we can call these analogous to, we use the term reaction center, but basically this is reaction center or photosystem 1, reaction center 1. And then this purple is different, right? We said they have the electron reverse flow. So one scenario is that these organisms had some sort of gene duplication, maybe a whole genome duplication. And over time, they ended up with a couple of different photosystems, but ultimately their original one was lost. So maybe this was the first photosystem that evolved. Then later on, these guys came into being in the current purples that we see today. But in another group, which led to the cyanobacteria, you instead ended up with a fusion then of what we see with these green sulfurs and purple sulfurs today into a single unified Z scheme. And that is what allowed, once you had two photosystems, then you could unlock the electrons from water. And so you can see why this may have taken billions of years to accomplish, because it would be a very slow process. So this isn't something we've said for sure is how it happened, but this is one scenario of how that may have evolved. All right, well, I have run over time, so I will end there. Any questions?